The scorpionfly, Panorpa vulgaris, is a conspicuous and fascinating insect within the species poor order Mycoptera that can be observed from May to September on low vegetation at forest edges. The insects are about 2 centimeters long and, unlike the true flies of the order Diptera, have 2 pairs of wings, which are distinctively spotted and held flat above the abdomen in a characteristic V-shaped position. As in all Mycoptera, the head capsule is elongated like a beak, which has given the order its German name Schnabelfliegen. At the end of the elongated frontal shield, the clypeus, there is a short labrum and hidden behind it, short, biting, chewing mandibles, whose teeth can be seen peeking out here on the left and the right. Underneath, along the entire beak, there are elongated maxillae with angular bases, and between them, hidden, an elongated labium, which seals the feeding canal from below. Both maxillae and labium have well-visible articulated pulps, typical for chewing biting mouthparts in insects. A feeding scorpionfly uses its beak to literally poke around in its food, usually dead insects. In this way, it extracts even the most inaccessible bits in a carcass, crushes them with the mandibles, and finally transports the liquefied food towards the esophagus. The long beak probably also helps them when stealing dead insects from spider webs, something scorpionflies are doing regularly and are very good at. Thanks to the beak-like head and the characteristic wing coloration, Panorpa vulgaris is unmistakable amongst our ID species. The males stand out in particular because of their modified abdominal end, which resembles the tail of a scorpion. This is of course not a venomous stinger, but the male copulatory organ with powerful claspers, which will be discussed later. Outside of our ID species, Panorpa vulgaris can easily be confused with other scorpionflies of the genus Panorpa, of which there are at least 3 other species in North Rhine-Westphalia. From Panorpa cognata and Panorpa germanica, Panorpa vulgaris can be easily distinguished by the wing pattern. Vulgaris has a dark band on the widest part of the forewings. Panorpa cognata and germanica have only single isolated spots. The wing patterns of the very similar sister species Panorpa communis, however, which often occurs in the same habitat with Panorpa vulgaris, are confusingly similar to vulgaris. They tend to be less heavily spotted, but this is variable. The only reasonably clear distinguishing feature is the basal patch, which in Panorpa vulgaris spans at least 2 wing cells, but in communis is either absent or spans only 1 wing cell, as shown in the picture. However, there is some variation in this trait as well, so that a reliable distinction of the 2 species based on external features is not always possible. Scorpionflies exhibit a fascinating mating behavior, which can sometimes be observed in the field. At the center of it all is the male's copulatory organ, which is armed with claspers. The end of the abdomen is bent upwards because the male stands by the side of the female during copulation and grabs her abdomen from below near her genital opening. The claspers enable the male to stabilize the connection during copulation and to transfer his sperm continuously during the course of mating. In addition, males have another morphological structure on the abdomen that serves the mating bond, the so-called nuptial organ. This is ultimately a clamp formed by the protruding edges of tergites 3 and 4. The forewing margin of the female is inserted into this indentation during mating. The female is thus fixed by the male in 2 places, at the end of the abdomen and at the forewing. But with a little effort, she is still able to interrupt copulation at any time. For mating to occur at all, the male must do a number of things right in a complex courtship ritual. First of all, visual and vibratory signals are used to lure a mate. For this purpose, the male beats his abdomen downwards at a regular interval, often accompanied by conspicuous wing movements. This behavior becomes more intense as soon as conspecifics are nearby, and especially when mating-ready females respond with similar movements. In order to initiate copulation, the male must then present a nuptial gift, a nutritious nuptial gift to be exact. The gift can consist of a dead insect, but even better are little spitballs, of which the male, here in the front, is just setting one down. These spitballs are drops of saliva that well-fed males produce in large quantities in their labial glands. The whitish, transparent drops are deposited by the males on the substrate, usually a leaf, harden quickly on contact with air, and are highly prized by the females. During copulation, which can last up to 2 hours, the female usually feeds permanently from the nuptial gift. When it is nearly consumed, the female becomes restless and makes an effort to break away from the male. In this case, however, the male is well prepared because he has already deposited a new spitball, which is quickly found by the female and snatched up from the leaf. Little is known about the exact ingredients of the spitballs. What is certain is that their consumption has a positive effect on the number of eggs that a female can lay, so they are real gifts with nutritional value. Notably, the females chew and lick on them for a very long time, given the small size, so they obviously cannot swallow them and be done. From the male point of view, that is desirable, because in scorpionflies the number of sperm transferred depends closely and linearly on the duration of the copulation. And since in Panorpa vulgaris females mate with many males whose sperm is mixed completely in the sperm receptacle of the female, the sheer number of transferred sperm determines male mating success. The males therefore do everything they can to ensure that the copulation lasts as long as possible. Once the spitball is over and done with after a few minutes, the next one is already in place and the nuptial pair turns another 60 degrees. Depending on the male's ability to keep producing new spitballs, copulation can last for 1 to 2 hours, with the pair spinning around in circles several times. I had said before that visual and vibratory signals are used in mate finding by Panorpa vulgaris, but possibly chemical signals, sex pheromones, are also involved, as they are in other species of Panorpa. In this courting male of Panorpa germanica, a species that also occurs in Central Europe, the protruding pheromone gland can be seen on the clasper segment. While releasing the pheromone, the male also literally waves around paired appendages on the clasper segment. In addition, it wags its wings, which is visually quite conspicuous, but may also contribute to the spread of the pheromone. In Panorpa germanica, the sex pheromone can actually be smelled. Reportedly, the smell is reminiscent of freshly cut cucumbers. Chemically, it consists of 2 short-chain, highly volatile aldehydes that have proven to be attractive to females in behavioral experiments. Panorpa germanica mating occurs only in the late afternoon to early evening and is much more rarely observed than in Panorpa vulgaris. Pheromone or not, also in this species the male has to deliver a nuptial gift, a dead insect or a spitball, to be accepted by the female. The males of our ID species Panorpa vulgaris have similar structures on the clasper segment, but the gland is not exposed to the same extent in courtship. It is therefore unclear whether pheromone communication is also involved in Panorpa vulgaris.